I thought I would start with something which you know much better than myself, which is, what do we understand by Parkinson's disease? Well, Parkinson's disease is classically defined by the problems of movement. It's defined pathologically by the loss of a very small subset of nerve cells in this bit of your brain. This is the stem of your brain. These are the hemispheres. And at the top of the stem, there's an area called the substantia nigra. And within the substantia nigra, you have 500,000 dopamine cells on either side of that structure. And when you've lost half of that, so 250,000, you develop the features of Parkinson's. So that's a pretty small population, given you've got something like 70 or 8 billion nerve cells in your brain. You only need to lose 250,000 and you get Parkinson's disease. So that is important to bear in mind, because that obviously has implications about how we treat it and the promise of what cell-based therapies can offer. Now, the treatment of Parkinson's disease I'm not really going to talk about, and the only reason for putting this slide up is to show that there are many good treatments out there for Parkinson's disease right now. And every single one of those treatments works essentially around making dopamine work in the brain, or deliver dopamine in the brain to treat those aspects of the disease. And as you will know better than I do, some people respond better to tablets than others. Some people do well. They have very little problems with thinking. Some people develop more problems with thinking and such like. So to set the scene for the work we were going to go on to do and are doing with cell-based therapies, we decided that it was important to begin with to make sure we understood what we were treating. So if you like, our hypothesis was that Parkinson's disease is not one disorder, but might be several disorders that look similar but behave rather differently. So this study, which was funded by the Parkinson's Society, as it was in those days before it became Parkinson's UK, helped fund this work. And I'm not really going to dwell on it, but essentially what we did was we collected everyone with Parkinson's disease in Cambridgeshire in 2000 and 2001, and we did it again in 2008 and 2013. So that gives us some community-based epidemiological data on what Parkinson's disease is like in the real community. Just some headline figures from that. The average age to get Parkinson's disease is 68. In Cambridgeshire, every 4 to 5 days, there's a new case of Parkinson's disease diagnosed. So this is a common disease. And we follow these people to the present day. And essentially what we found is that there are a group of younger patients who are very good at thinking, have no problems at all. And there are a group of people who tend to be older, and older in our group was over 72, who had some problems with thinking. And these patients who had these subtle problems with thinking sadly seemed to go on and develop major problems with thinking early on, which led to a dementia in their particular case. And these two types of Parkinson's disease seem to be rather similar. They also seem to have rather different genes behind them. So there were genetic variants, things which you're born with. These are not genes which you inherit, which cause you to have the disease, but change how the disease behaves. So our thinking has been that what we have here is we have a form of Parkinson's disease that affects younger people, may have subtle problems with thinking, but that's not a major problem. They don't have these particular genetic variants, and they tend to do very well, progress very slowly, and their disease really targets predominantly the dopamine nerve cells that I've talked about. In contrast, there is a group of patients who tend to be older when they get the condition. They have subtle thinking problems when they first present to their GP, which relates to their age and certain genetic background. And these patients, unfortunately, have a more malignant form of disease where it tends to gallop away a little bit with them. Now, that's important, and that's important because whatever my therapy is, so I'm going to come on and talk about cell-based therapies making dopamine. If that is true, you can see the problem that could arise if I don't take that into account. So I make the best dopamine cell therapy in the world. I give it to 10 patients here. I give it to 10 patients here. I wait 3 years, and at the end of 3 years, I say to the patients, do you think you're any better for that therapy? These 10 patients who have a disease which only really affects the dopamine cells will say, I feel fantastic. I feel absolutely crackerjack. I feel that therapy's changed me dramatically. Sadly, this group, 3 years down the line, the dopamine problems aren't the major problem now. They've got problems with thinking. Things are not going very well. I'll say, how's the cell therapy working? They'll say, it's dreadful. Hasn't helped me at all. In fact, I've got worse for your cell-based therapy. 10 have got better. 10 have got worse. I put them together. The conclusion is, doesn't work. 10 were good, 10 were bad, doesn't work. End of therapy, end of experimental medicines for Parkinson's disease. back to the 1960s. And that has been a dominant problem in this field, that people have not taken that into account. So this understanding of the fact there may be two types of Parkinson's disease has implications for how we roll out the next generation of therapies, including cell-based therapies. This group of people will do extremely well with therapies which are designed to replace dopamine with cells, replace dopamine with genes, which I'm not going to talk about, make the dopamine cells grow better with fertilizer, GDNF, this glial cell line-derived neurotropic factor that Arthur was mentioning. All this will be good for this type of patients. What about these poor patients over here? These patients are ideal for the other new therapies that are coming along, which are designed to slow down the disease. And at the end, I will mention one possibility which is very exciting, which is the idea that you might be able to use vaccines, immune therapies, to kill off the protein that causes Parkinson's disease. And if you wanted to trial that, this would be the ideal group to do it. So just understanding the fact that there are these two types of Parkinson's disease has important implications therapeutically. So this is the group I'm really going to talk about, where we should be targeting these therapies in the first place. So just to sort of recap at this point, we think Parkinson's disease probably is one disease, but it's two types. One runs very slowly, tends to affect younger people. There's an older version of it where people tend to do less well. That younger group in particular are ideal for our new experimental therapies, but it doesn't mean it can't be rolled out to more patients as we get proof of efficacy. So the rationale, just to get this absolutely clear with our cell-based therapies, our rationale with this is not to cure people of Parkinson's. This is not getting rid of the problems with Parkinson's, it's just a better way of treating it. So that loss of those dopamine cells is what we're trying to do with our cells. We're trying to replace those lost dopamine cells. And we know that should work. And why do we know that should work? Because when we give people dopamine drugs, they do fantastically well for many years. So there is absolutely no reason to believe that if we put dopamine into the brain with cells, it wouldn't work as well. So that's what we're going to try and do. Are there any advantages with doing that, outside the fact we're just putting dopamine back into the brain in the form of a cell? Yes, there are two major advantages to that. One of the problems with the dopamine drugs we use at the moment is when you swallow a dopamine tablet, it obviously works on the area where you haven't got dopamine. In those nigral dopamine cells which project up to the brain, you lose dopamine, it replaces it. It will, of course, replace or work on dopamine systems which you have in other areas of your brain. So one of the problems with some of the dopamine drugs is they change people's behavior, they can make people see things, they can make people slightly confused, and all of that is because dopamine is working where it's not really needed. So this has the advantage that you can put dopamine into the bit of the brain where you want it to work. The second advantage is if I put dopamine back in the form of a nerve cell, that nerve cell will release dopamine just like a normal nerve cell. When you take a tablet, it gets taken up in the gut, gets taken up into the brain, gets taken up into the nerve cells, and then it's just released. It is not released in the way in which dopamine is normally released. And that has the consequences that after a number of years, the tablets start to behave in a slightly erratic fashion. Sometimes they don't work, and people develop the involuntary movements, the so-called L-DOPA-induced dyskinesias, which you see in people on long-term therapy, which would be avoided by this therapy. So what we are hoping to do with this is to put dopamine back where it's needed and have it released in a way in which the brain normally releases dopamine. And by so doing, in theory, we should remove the need to take dopamine tablets, and you would then remove the need for all those other therapies we have when people have complications from it, such as deep brain stimulation. So that's the rationale, and it's important because a lot of people think we're trying to cure the disease. We're simply trying to give a much better, sustained, targeted delivery of dopamine into the brain. So where have we been? This is not a new concept. This concept actually came about in the 1980s. So if you want to take a cell therapy from an idea through to a therapy in patients, you do it in a certain way. You obviously have to identify your dopamine cell. You then have to test it in an animal model of Parkinson's, which I'll come back to. If that looks successful, then you try it in a small number of patients to see whether it's feasible and whether it's tolerated. Then you try and work out how to do it better, do it in larger numbers, and eventually you end up with the gold standard, which is a double-blind sham surgery trial. These are trials where people either have the treatment or don't have the treatment, but the patient doesn't know, the doctor doesn't know, and therefore everyone is blinded to whether they're actually having the treatment. And that is the final stage in the development of your therapy. And it's important to remember it's the final stage because you will come to see that other people haven't seen it in terms of this sequence. So what have we managed to do in this area? Well, anything you can think of that could make dopamine has been tried. So this is an area that attracts a lot of excitement and a lot of madness. So there are lots of crazy things that people have done, and you will see later on one of those slightly crazy ideas. But the one therapy, the one cell that really seems to have worked well are when you take the dopamine cells out of the developing fetus. So those dopamine cells, which I was telling you about in the nigra, in the substantia nigra in the adult brain, develop in an area called the ventral midbrain, the ventral mesencephalon in the developing fetus. And if you dissect out those cells at the right age and you transplant them in, you can get dramatic effects. And in the animal models, what we do is we use this model. Now, the only animal on the planet that gets Parkinson's disease is man. So no animal has Parkinson's disease. So we cannot mimic Parkinson's disease in the lab, even today. So what do we do? Well, what we do is we knock out the dopamine system with this toxin. So what we're actually modeling is loss of dopamine. But that's what our therapy is designed to do, so it's perfectly reasonable. And so in the 1980s, they got Mr. Ratti. They knocked out dopamine on one side of his brain with this lesion. They took out the developing dopamine cells from the rat fetus, transplanted it in, and here's an experiment I did many years ago, now 20-odd years ago. And all this brown you can see on this slide are the fetal dopamine cells that I've transplanted into the adult rat brain. So all these dots are the dopamine cells that have survived, and this brown halo are all the fibers that have grown out. So using this simple model, you kill dopamine off on one side of the brain, you transplant in the developing dopamine cells out of the developing bit of the brain, they survive, they make connections, they receive connections, they release dopamine, and Mr. Ratti gets better. Now, I mean, that's pretty amazing when you think about it, because you're actually taking out a bit of developing brain, which is essentially dead when you're taking it out. You put it in the brain, the cells survive, and you get these sort of dramatic effects experimentally. So that's what happened in the early 1980s. So to recap, if you use the tissue from the right species, so the same species, so this was rat to rat, so when you're going to take it to the clinic, you're going to have to get your fetal material from the human, so that's going to involve abortions, and that brings ethical problems with it. You have to get it at the time the dopamine cells are developing, so in the human, that's 6 to 8 weeks old fetuses, and it only works if you put it where dopamine normally works, not in the substantia nigra where you lose the cells, but higher up in the brain in the striatum where dopamine is lost. So if these experiments are right, then if I do the same in the human Parkinsonian brain, if I take human fetal tissue of 6 to 8 weeks, put it in the striatum, I would expect it to survive, I would expect it to make connections, I would expect it to release dopamine, and I would expect the patient to get better. That is my preclinical data, which gives me the confidence to go to the clinic. So in Sweden, in the late 1980s, this is what they did. First 2 patients they did made no difference at all. Patient absolutely no different. And they went back and thought, well, perhaps we haven't transplanted enough and perhaps we should change the way in which they did it. So they did. And this is patient number 4, who became one of the most famous patients in the world with a transplant. So in 1989, this patient had a transplant on one side of the brain with the dopamine cells collected from 4 aborted fetuses. Prior to having the transplant, that patient spent 2/3 of the day off. So for 2/3 of the day, despite taking all the drug therapies that existed in the late 1980s, they essentially were really unable to move. So for every 8 in 12 hours, they couldn't really move. They had a transplant placed on one side of their brain, and for the first few months, nothing happened. 3 years after they had that transplant, they had gone from being off 8 hours a day to being on all day. So they had no off time at all, which is a pretty impressive response. It's even more impressive when you realise that over that 3-year period, they stopped all their medication. So 3 years after a transplant on one side of the brain, they were on all day, they were on no medication. So they were clinically better, their behaviour had improved. These patients came over to London and had a scan done at the Hammersmith Hospital where they looked at dopamine in the brain with a particular type of scan. Here is the brain. This is the bit which particularly is lost with Parkinson's, a thing called the putamen. And as you can see, as this patient improved over 3 years, at the site of transplant, the graft got brighter and brighter as it matured into dopamine cells. So they clinically got better, and that correlated with the dopamine cells growing at the site of implantation. The more vigilant will also notice that on the non-grafted side, so the side that did not receive the transplant, the dopamine cells have continued to die off, showing that the disease process is continuing. Now, this was then taken on by groups in America, and one of the patients in America, 18 months after they had this transplant procedure, had a heart attack and died. They had improved, their scan had improved, and when they looked at the brain, these were all the dopamine cells within the transplant, and they were sending little processes into the brain of the patients with Parkinson's disease. So just as in the preclinical animal studies, we could show that they got better, the patients, and that they transplant survived. Now, patient number 4 came back to the Hammersmith a number of years later and had a different type of scan where they stimulated the scan that the patient's transplant to release dopamine and showed that it released the chemical you're interested in, dopamine, just as the normal brain. So it released dopamine exactly as the normal brain did. So these experiments, if you like, demonstrated in the patient that what you had found in the animal, you could do in the patient. So then the question is, well, why are we all sitting here discussing it now rather than you've all had this therapy? What is the problem? Well, the problem you can already see here is that these 2 patients, I've concentrated on patient number 4, and patient number 3 didn't do quite so well. Now, why did patient number 3 not do quite so well? They clinically didn't improve, their scan didn't improve. So what this told us was we still didn't know how to do it properly. So we were still trying to work out how best to do this. Now, in America, what happened was, in 1992, was that George Bush Sr. Left the White House and Bill Clinton came into the White House. Now, what was the importance of that? Well, the importance of that was that Bill Clinton, as one of the first things he did in the White House, was allow federal funding for fetal tissue to be used by researchers, whereas previously it had all been done in the private sector. So the National Institute of Health, who were very excited by this data, put out a call saying, should we look at fetal tissue transplants in Parkinson's disease? And they funded 2 trials at this point. So at the point at which they funded 2 trials, in the published literature, there were about 12 cases of people who'd had a transplant. And those 2 trials were these double-blind, placebo-controlled trials. So these are trials where people either have the active treatment or they just have pretend surgery, a pretend transplant, and the patient nor the clinician who's assessing them knows whether they've had the real thing or not. Now, there are lots of ethical questions around that. The big problem we had was that this was being done at a point when you didn't know what you were doing. So to take this to its extreme, if in 1953 I did a similar experiment, I say, well, actually, there's these funny things called antibiotics. They look quite exciting. Let's have a go with these. So I take a group of patients, 40 patients. I give 20 of them 100 milligrams of penicillin, and I give the others a pretend tablet. I do a chest X-ray at 5 days, and I say, does penicillin kill the microbes that cause chest infections? And what you'll find is it didn't work. Now, why didn't it work? It didn't work because all the patients had different chest infections. It didn't work because you used the wrong dose of antibiotic, and you only waited 5 days to clear a chest infection, which is too short a period of time. So that trial would have failed. The conclusion you can come to is that penicillin doesn't work for the bugs that give you chest infections. Therefore, antibiotics don't work for microbes. Let's abandon antibiotics and go back to putting people in clinics up mountains to treat chest infections. Now, it's an extreme example, but you can start to see how the logic starts to unravel, that when a trial fails, the assumption is it's your agent that's failed rather than your trial failed. So from what I've said already, you can start to see why this might fail if you haven't taken into account the type of patient, if you haven't taken into account how you can actually do this optimally. Now, as you can imagine, these trials, which both took very different approaches, didn't work. And this led to this lovely headline in The Guardian in 2001, which also personifies what happens in the popular press. So in the popular press, there are only ever two outcomes with any medical intervention. It's either miraculous or catastrophic, because that sells your paper. So you could not say, new trial in Parkinson's disease, which had some methodological problems, produced some interesting but also disappointing results. No one's going to buy a paper that says that. But that's a much more honest account of it. So what were these two trials which produced this disastrous and catastrophic result? These are the two trials. I'm not going to dwell too much on it, but essentially in this trial, 40 patients were taken to theatre, 20 had a transplant, 20 had a pretend transplant. After a year, you were phoned up and asked if you were better. That was the trial, nice and simple. And the patient said, no, I'm no better. And there's all sorts of problems with that, which we can discuss, but essentially there was no better, they were no better. And some of the patients, which was the worry, had now developed some of these involuntary movements, these dyskinesias, which you see in nearly everybody after they've been on L-DOPA after a number of years, but now you were seeing it when they had a transplant. Now, of course, I can get rid of anyone's L-DOPA-induced dyskinesias because I can get rid of their L-DOPA. If you've got a transplant-induced dyskinesia, I cannot get the transplant back. It's in your head and I can't get it back. You could treat it with deep brain stimulation, which had now come along, and that works quite well, but it was a side effect. In the second trial, which was published 2 years later, patients were randomized to have no transplant, small transplant, big transplant. They waited 2 years, they did a standard measure, and they showed whilst there was a clear trend for benefit with the transplants, it was not statistically significant, and patients had now developed these involuntary movements. So this seemed to suggest that this, what I've told you, is all rubbish, doesn't work, and produces side effects. So if that is true, how could you explain this? So this is a patient that was looked after by Neil Quinn in London and is looked after by my colleague Tom Faltney in London. This is a man who's had Parkinson's disease for 30 years. Those are his graft-induced dyskinesias there, those leg movements. He was transplanted 20 years ago and he's on no medication. And there are two of these patients that have been followed up in London long term. They have motor scores that are less than when they presented to their doctor 30 years ago, and they're on no medication for their Parkinson's disease, and that's what they look like. So it's pretty hard to believe that those American trials were absolutely right in the conclusions they came to when you saw that. So of course you now have a problem, especially when stem cells are coming up and looking to go to the clinic. Is this worthwhile pursuing? Do cell-based therapies work? Who's right? Are the Americans right? Or are the European open label studies right? So of course you can pick your side. There's a certain bias, I'm sure, in this audience as to where we're going. And of course you can just then shout at each other until whoever shouts the loudest wins. But essentially what we did was we went back and looked at all the data. And when you start to go back and look at the data, all the points I've raised, you can start to see the problem. So you're comparing these two American studies with the studies in Europe. So can you do that? Well, the answer is you can't. And why can't you really compare them? Because none of them took account of the type of patient they were actually transplanting. They all used different types of patients based on what I was saying earlier on. They all used different doses of cells. So some used small, some used a lot. None used as many as were used in the European studies. Some were delivered from the top, some were delivered from the front. They were delivered in different ways. So it's like taking a tablet orally or someone injecting it. Some of the patients weren't given any immune therapy to stop the transplant being rejected. So some had immune therapy, some did not. Some gave it for 6 months, some gave it for 2 years. So protection of the graft was very different. They used different endpoints. Some phoned them at 1 year, some used measured at 2 years. The Swedish open label study just carried on forever. And one of the greatest sadnesses in the American studies, they were funded for 2 years to follow up their patients, At 2 years, you finished. So if any one of you was in those studies, you were out of the study and you just disappeared back to wherever you were in America. So out in America at the moment, there are patients who've had transplants that nobody is following up. So how can we reconcile that? How could we use that information? So where do we go next? So what you do is you go back at the data and say, well, is there a group of patients that are going to do well? So I've already told you a little bit about that. So we went back and analysed all the data, and what we found was that the patients who did best in the trials to date, even the American trials, tended to be younger patients who were less advanced. So patients who had had disease for less years. The problem was these involuntary movements that I've told you about. What was causing those? Why were they developing in people who'd had transplants? And there were two theories. One was that the transplants had meant that you had little hotspots of dopamine in the brain, so they hadn't been delivered properly into the brain. You had hotspots in areas where dopamine was really overactive and other areas where it was not working. And the other was that what you'd also transplanted were a group of nerve cells that mishandled dopamine, so-called serotonin or 5-HT neurons. So what we did, or what you can do to try and make sure that won't happen, is that you deliver the cells evenly across the brain and you avoid these contaminating cells. So you can go back and analyse data and say, I think I've got an idea who's going to do well, and I think I know why patients develop these side effects. So you can then use that information going forward to plan a new trial. And so that brings us up to the present day. That's the past. We had this mixed history of what happened, so it's moving forward. Now, I have to say that Parkinson's UK were pivotal in this work, because in the early part of this century, the perceived dogma was that these transplant trials didn't work, and everyone was wasting their time thinking about even doing another trial or taking it forward. We tried to get everyone together to analyse that data, including the American groups, and Parkinson's UK funded these people to come over to from America. So we had meetings every 6 months or so in head office down in London to put together what was to be the next trial. And that next trial was Trans-Euro, which I have to say I thought was a brilliant title, because if you apply for European grants, you have to have a very snappy title to your project. And the great thing about Trans-Euro was it sounded as though that all of Europe was involved, which I knew they would love, but it also captures the transplant part of this. So this is a study which we put the grant in for 2008. And it involved Cambridge here as the coordinating centre, which was bringing together patients from London and Cardiff, Paris, who never really got going, Vienna, who looked after the ethics, a site in Germany, and our good friends in Sweden, in Lund. And that set up Trans-Euro. It's a €12 million grant, and it was awarded in 2010. And what we did was we selected the patients we thought would do best, using what we'd found before, so younger patients, less advanced, with very little in the way of L-DOPA-induced dyskinesias, and we collected 150 of these people across Europe who get followed up every 6 months. We optimised how to prepare the tissue in terms of making sure we had the right number of dopamine cells and none of these contaminating cells, and we worked out how to transplant it so we could get the cells evenly distributed across the brain. And that took a couple of years to sort all that out. And then what we decided to do was that how the trial would work was that all these patients would be assessed every 6 months. 40 of them would be randomly selected to go and have some expensive imaging in London, that PET imaging. 20 of those would then be selected to have a transplant. And that would mean at the end of the trial, we'll have 20 transplanted patients, another 20 who've had the same imaging with the expensive scanner, the PET scanning, and another 110 who've been followed up in exactly the same way. We will wait 3 years to decide whether it's really worked, and we use a number of measures, and importantly, everyone wears a baseball cap when they're being assessed. Now, why do they wear a hat when they're being assessed? It's not a fashion statement. It's there so we can video the patient, and then we can send the video to someone else, and that person will have no idea whether that patient's had a transplant or not, because they're wearing a cap, so they can't see whether they've had surgery. So that person can then assess the videos, not knowing whether the patient's had a transplant or not. So that blinds at some level against the bias. And we are currently here So we transplanted our first patient in Cambridge in May of last year, and we've now done 10 transplants. So 5 patients have had transplants on both sides of their brain. So we've done 5 bilateral transplants, and last Friday, so however many days, that is 4 days ago, the first patient in Sweden had their transplant. So we've done 11 transplants in 11 months. Now this is a problem using fetal tissue. There are ethical problems, there are also logistical problems, which is why this trial, setting it up, was always seen as a stepping stone to where we go next, which is with stem cells. And that's where we are now. So TransEuro is active, it's running, and we hope to finish the transplants this year. The future, however, are stem cells, because they are, first of all, well, ethically less contentious. I think that is still contentious in itself. There are major advantages with using stem cells in that you can grow these cells in the lab, you can get large numbers of them, you can package them up, and trying to get hold of the tissue won't be a problem. To do a transplant for a patient with fetal tissue, I have to collect at least 3 to 4 fetuses where I can dissect out the bit of brain I want, keep it alive for 4 days in a particular media, and then transplant it. I think we've cancelled something like 17 operations because we've not had enough tissue. So that's a major logistical problem. So that is why this is a very useful tissue in terms of the logistics. The question is, well, which stem cells are we thinking of? And there's tons of these different types of stem cells. So, of course, we've all got stem cells in us. Our blood is being replaced every 120 days by stem cells. So there are lots of different stem cells within the adult body, and there are those which obviously make up us during development. But I would say there are essentially only 2 types of stem cell that are going to go to clinic in the foreseeable future. And if you read about any other stem cells for Parkinson's disease, you should ignore it, because there is no evidence that any of those are going to work. So what are the 2 stem cell sources which I think will go to clinic? The first are these so-called iPS cells. So these are cells which is an invention or a discovery that was made in 2006 by someone called Yamanaka, and he won the Nobel Prize for this in 2012 with our very own John Gurdon here in Cambridge. But what Yamanaka discovered was that if you take an adult cell, so if I took one of you here, I could take a bit of skin. I take a bit of skin and I grow the cells out of the skin called the fibroblasts. I then take those fibroblasts and I chuck on a certain number of factors, and I then convert those fibroblasts back to where they began in life. They go back into these pluripotent stem cells, the cells which give rise to every cell in your body. So I've de-differentiated them back to this so-called induced pluripotent stem cell state, and then I can drive that into a dopamine cell. So in theory, I can take a bit of your skin, grow up the fibroblasts, turn them into iPS cells, turn them into nerve cells, turn them into dopamine cells, and then I can treat you. So that sounds quite exciting, the so-called iPS route. It works in rats, so Mr. Ratty, going back to those experiments I was telling you about at the beginning, I can do it, take a bit of rat skin and do exactly what I've said. You can do it in monkeys. So in Japan, where they invented this is what they're really investing in. You can do this. And then more recently has been the idea that you can take the skin cells, you can make the fibroblasts, but rather than turn them into stem cells, you can directly turn them into nerve cells by making them induced neurons. So that's very quick, because that works very quickly. So now I can take your skin cells, grow them up in the lab for a couple of months, chuck on a different bunch of factors, and I've got dopamine cells in a week. So that's pretty exciting, and you can do that in Mr. Ratty as well, or mouse in this case. The question you then ask is, that's terrific. I mean, to do that is not straightforward. The cost of that is prohibitive. And ultimately, you'd have to ask yourself, if I take your skin cells, turn them into iPS cells or directly into dopamine cells, but I make dopamine cells, do you really want them back? Because you've got Parkinson's disease, so your own dopamine cells weren't doing so well. So if I'm going to put new cells into your brain, do you really want the cells from you, which might have some genetic problem as to why they didn't live for 100 years in the first place? So I have problems with this conceptually, but there are real issues in terms of the practicalities of it. So that means, I think, we're really going to have to work on these so-called embryonic stem cells. So these are the cells which come from the spare embryos from IVF. So when the egg's fertilized, it divides. Once it's divided a certain number of times, you end up 16 cells, and these are the embryonic stem cells. So every single person in this auditorium was at one point in their life an embryonic stem cell. Once you've got an embryonic stem cell, because it gave rise to every cell in your body, you now want to say, can I persuade the embryonic stem cell which I've got to turn into a dopamine cell? And in the last 5 years, that has happened. So by a group in New York and a group we work with in Sweden, they now have a protocol which allows you to make dopamine cells incredibly reliably from human embryonic stem cells, and that they actually work when you transplant them into the rat brain. So going back to those original studies, we can now take embryonic stem cells, we can turn them into dopamine cells, we can transplant them into our animal models, and they work. And they work as well as the fetal tissue when we do those transplants. So we're now at the point where we can make, if you like, from a 6-well plate, it's about that big, I can make enough with embryonic stem cells in it, I can make enough dopamine cells from that to graft 5 lecture theatres like this. And I can do that with a protocol that takes 2.5 weeks. So that is there, we can do that. So where do we go next? Well, the challenge is, how do we do that? How are we actually going to do that? And one of the things we've tried to do is we've tried to join up all the groups in the world who do this to make sure we do it properly and none of us mess it up like it happened before. So this is a thing which we set up with the Americans and the Japanese and the Europeans. And all of these groups feel that they will be filing for clinical trials in the next 2 to 3 years. And I personally think we will be doing a first-in-human embryonic stem cell dopamine trial in Cambridge in 2018. Now, not only is it important to set up these collaborations to allow us to work towards delivering a clinic at the right time, it's also important that these organisations can then assess the other claims that are out there. So some of you may have seen this. This went pretty viral just before Christmas. So this is a company in America which are doing a trial in Australia. So that should instantly raise queries as to why they're doing that. And this company have got a parthenogenetic stem cell. So this is where they haven't fertilised an egg. They've taken an unfertilised egg and made stem cells out of it. And they use that then to say, we're not destroying an embryo, so this is an ethically more acceptable source of cells. They have got ethical permission. They've got regulatory permission to do this in Australia. They've now got ethical permission from the hospital where they're going to do this, and they're going to graft 12 patients and follow them up for 12 months. If you look at the papers they cite supporting the use of this, all those animal experiments I told you about, however simplistic they are, have not been done. None of the data shows that they can make any dopamine cells out of these cells. So there is no preclinical data supporting the use of this, and yet it's going to a clinical trial. So we wrote an article, which is freely available in the Journal of Pug Disease that came out 2 weeks ago, questioning this whole approach and trying to get people to critically appraise this type of data. Because if this goes wrong, this trial, it's not only bad for those patients, this will be the end of the field. People will say it's a dangerous field, it doesn't really work with the fetal tissue, this is a major problem, and that will be it. So all that work will be wasted. So it's very important that I think as a patient organisation, as patient charities, that you actually critically appraise this and make people aware of some of the anxieties you have with these trials. Our plan is to use the protocols we've said. We hope to recruit the patients who are still in the transurose study. Those who haven't had the fetal transplant, we will graft with our dopamine cells. We will do exactly the same assessments that we've done already. We will have to do a few more to check on safety. And the cell we need to prepare is where the problem lies at the moment, because this is a very complicated process. We have to guess the number of cells we want to use, and actually there are lots of regulatory problems we have to work out. We have to work out what's the best cell line to use so we can actually use it in everybody we want to. Can we actually do it at the levels which are required to be used in the clinic? Can we make sure we're only putting in the right cells? Can we make sure we can store them safely? Can we use them anywhere in the world, because there are regulatory chain differences? So what you can do in Europe with embryonic stem cells is different to America. America's different from Europe. And we need to know exactly what we're going to transplant in terms of the criteria. So the detail is important, but that's what we've got to do. So that's the last 2 years' work, next 2 years' work, is just to resolve those final issues. I don't think there are any problems. We just have to do it for the regulatory necessity. So ultimately, in 2018, we will take patients who we already know, we will put in the cell product that we think we can make, we know how to graft it, we will slowly increase the dose, we will use exactly the same strategies as we've done before, and that will enable us to show whether this therapy will really work in Parkinson's disease. And is that the future? So in 5-10 years' time, will everybody be having this therapy? Well, it will only ever become a therapy if you can make it competitive. So what does that mean? It means that it doesn't just have to work. So if it makes you a bit better, that's no good. It's got to be better or as good as the things that are out there. So that means you've got to improve dramatically with these therapies. So you're going to have a big sustained response. We've got it with fetal tissue. No reason we won't get it with this. You've got to have side effects that aren't a big deal. Well, we're not putting in the cells that cause the dyskinesis with the fetal transplants. We can deliver it to brain. I don't think that will be a big problem. What about the cost? So what will this ultimately cost? So what does it cost to treat Parkinson's disease? Well, these are some figures I put together. So if you look over a 10-year period, L-DOPA costs about £10,000 over 10 years. If you go for a standard dopamine agonist or a Pinerol, it's 3 times more expensive. Apomorphine, 8 times more expensive. Deep brain stimulation actually is not very expensive. It just costs an awful lot to have it in the first place, which is what everyone sees. But over 10 years, it's not too bad. Duodopa, where you have it put in the small bowel, over £250,000. So that is serious bucks for that. For our transplants, it costs £23,000 to have 2 transplants and a year of immunotherapy. So our stem cell transplant will cost that just to deliver it. So the big unknown will be actually who's going to ultimately own this. Because when I finish my phase 1 study, we do a phase 2 study, someone's going to have to invest in it and buy it and take it. And ultimately, whoever buys it will determine the price. And that, I think, again, is where the patient groups can be so important about if this really works, making sure that the price doesn't become something which stops you from having it. And the final thing I'll say before I wrap up is that this whole approach has told us how we might be able to repair the brain with Parkinson's disease using cells. We're at the exciting point where we're about to move forward with stem cells. One of the observations that's been made in this field is that when some patients died 10 years or more after having a transplant from unrelated causes, they looked at these transplants, and what they found to their amazement was that these fetal dopamine cells, which are only 10 years old, have got Parkinson's disease in them. Very small numbers, didn't affect how the transplant worked. But the question is, how could a transplant of such young dopamine cells have acquired the Parkinson's disease pathology? And so this raised the idea that the protein which causes Parkinson's disease, alpha-synuclein, has got into the transplant and started to cause problems inside the transplant itself. So that the pathology of Parkinson's disease has spread and infected the transplant. And if that's true, that the alpha-synuclein, the protein that lies at the heart of it, is spreading, can we stop that spread? And that has led to this huge revolution in the idea of using vaccines and immune therapies to kill off alpha-synuclein and stop the disease in its track. Now, none of these are mutually exclusive, so you could see in the future people come along with Parkinson's disease, have a transplant, have an injection of a vaccine. Thank you very much. I'll disappear now and won't trouble you with my neurological problems ever again. So to finish, where are we with stem cell therapies? I think we need to understand that this treatment is not for everybody, because we need to be careful how we use it. But if we do that, we will get much better results and know how to go forward. There are lots of new therapies that are out there, but we are getting to clinic with stem cells, and they will be coming to clinic, I'm quite sure, in the next 2 or 3 years. You need to be aware of what's out there. If you want to have a stem cell transplant for Parkinson's disease tomorrow, there are several hundred clinics around the world which will offer you that. They will liberate you of somewhere between $5,000 and $25,000, and there is no scientific basis for it. So this is a field which is fraught with danger because people are not doing what I've tried to describe tonight. And there are new therapies that come along, and these new therapies, which were new in the 1980s, which we're still working with, have thrown up unexpected results which have totally changed where we've gone with respect to our thinking on Parkinson's disease.